All righty. Let us go ahead and come on. I don't know why the first click never works. All right. So today we're going to continue our discussion of prokaryotic cells, and specifically, we're going to be talking about cell transport today. All right. So here's what we're going to do this today. We're going to cover this week's learning objectives. I'm going to open it up to questions. We'll talk about diffusion and osmosis, the structure of the cell membrane, and transport in and out of the cell. Okay, and then we will wrap up with what to do for next week. Okay, so this week will help you achieve the following learning objectives. Define the key role of evolution as it applies to microbiology. Identify microbial structures and connect the structures to their functions. Identify pivotal components of microbial systems important to human health. Okay. All right, so any questions? Any questions you have from your reading?
Teresa,
Yes.
Last class you mentioned something about in the questions for the test that you wouldn't put, you said something like you wouldn't put the answer in the question, or you won't word it a certain way. Can you re-explain that for us?
Oh, okay, sure. Let's go to, let me think of how to go over that. Tell you what, I will end this PowerPoint and I will go back to the one from last time. And the question that I was referring to. So let me scroll down and let's get to that. Okay. So let's go ahead and not from the beginning. Let me try this again.
Because the guys are fixing the.
I clicked the wrong thing. There we go. All right. So for questions like this is a good example of a question that you would see on the test. Not this particular question, but working your way through it. Okay. So when we have a question like this, why do we consider viruses acellular? Why do we use that term? The correct answers, there's two of them, and on the test, unless it's a multiple answer question, I will try not to have two correct answers, but during class it's good for discussion. Okay, so they are incapable of reproducing, or no, they do not have an independent metabolism, and they do not have a cell membrane. Those, okay, are the correct answer. Like I said, unless it's a multiple answer question, I would have just one. And if after the test closes, during my office hours, I'll have it set up so that you can come and talk to me individually on Zoom. And what I'll do is, if you want to, we can go over your exam. And if you say this was a multiple choice question, and I said this was the correct answer, but you picked this one, if you can make a good case that this is also a correct answer, I'll go through and give full points to everybody for that. Okay.
Coffee for the program.
Thank you.
Yeah. Oh, quite welcome. And then one thing and then this of course is not a correct answer, because viruses can make more of themselves. They do replicate, kind of reproducing. But then this question, they are not composed of cells. You want to watch out for answers like that, okay, that are true statements, but they don't answer the question. Okay, they don't answer the question of why we consider them acellular.
Yeah.
So It's basically just restating the question.
It's a
not
birthday party, it says. A birthday party? That's what it said.
Will they be similar to what we see on Cerigo?
Yes, there will be some questions that are Cerigo questions that I've pulled directly out of Cerigo. So it'll have the definition, and then you'll have a series of answers. Excuse me, let me back up. It'll have the term, and it'll have a series of definitions that are straight out of Cerigo.
Okay,
So there'll be a certain number of those.
all right, thank you.
Yeah. And then there'll be other questions where I expect you to know the Cerigo terms, and they're embedded in the question. Okay, did that answer your question?
Yes, thank you.
Other questions? Okay, if you think of some, let me know. I'm happy to answer them. Okay. Speaking of things on the last PowerPoint that I didn't get to in this class, and also forgot to mention yesterday, make sure that you know about endospores. We're going to be talking about endospores a lot through the course of this class. Okay. And so the information in the book is very good. I also have some recorded lectures from many, many years ago that are in the study aids that you can go and look at that. Okay. All right, let's talk about diffusion. Okay, this is going to be going back to chapter 2. And you're going, oh no, I hate that chapter. But you know, it's important for talking about transport of nutrients in and out of cells, transport of waste products in and out of cells. Okay. All right. So if I've got a beaker, okay, and I've got a beaker of water here, and I drop a sugar cube in, and we're going to say this is a colored sugar cube, okay, so that we can see it start to dissolve. Okay, so just as a reminder, why is it dissolving? The sugar, which is a carbohydrate, is what? Hydrophilic.
Philip.
Very good. Okay. So because it's hydrophilic, okay, the sugar is going to hydrogen bond with the water in the glass, and it's going to start to dissolve. Okay. And to start out with, the number of sugar molecules is going to be highest around the sugar cube. Okay. And these sugar molecules are going to run into water, and they're going to rebound, hit other sugar molecules, and they're going to bump out, and they're going to zigzag and just randomly, okay, move as the molecules bump into each other. Okay, and I haven't drawn the water molecules, but they're in there getting bounced around. Okay, so the analogy that I use that we're familiar with, okay, it's kind of like being on an elevator. You're all crammed in there. That's the sugar cube before we put it in the water. Okay, we're all in there. Okay, you may be bumping into each other, but you know, there's a limited amount of space that you can move around. Once the doors open, we go out, we spread out. Okay, and that's what the sugar is doing in the water, is it spreading out. Okay, just from randomly bumping in to the water molecules and each other. And I'm going to go ahead and put the water molecules over here. Okay, I should put them over here, just so we do it clear. Okay. And eventually, those sugar molecules are going to randomly spread out and fill the container of water. Okay, And it's because they're still randomly bouncing around. Okay. So this process, okay, of solute, okay, solute is the stuff that's dissolving in water, which most of the time is the solvent. Okay, so this movement of the solute, in this case sugar, moving from an area of high concentration to an area of low concentration, okay, until we reach what is called equilibrium. Okay, so it's equally spread out. Okay, so going from high concentration to low concentration, okay, going from high to low, okay, is called going down, down the concentration gradient. Okay, so down is not necessarily down. Going from high to low is down. Okay. Going from low to high is going up the concentration gradient. Okay, so going this direction. Okay, so if something randomly bounces from here heading back this direction, it's going up the concentration gradient. But it's less likely to do that, okay, because it's got all this space to go into. So it's kind of like when the elevator doors open, it's me stepping out and turning around and going back in. Oh, not my floor. Okay, it's harder because all the other people are coming out. Okay. Okay, how are we doing on this going up and down the concentration gradient? Okay, we call this diffusion. Okay, going from areas of high concentration to low concentration until we reach equilibrium. Okay, now you can play fun games with diffusion with scent. Okay, like if you have an orange and you have somebody in the same room with you, when you peel that orange, okay, you have that lovely orange scent very quickly diffusing into the air around you. So you smell it first, but eventually someone further away from you is going to smell it because those orange scent molecules are spreading in the air. Okay. You can do it with unpleasant odors too, but we prefer to go with pleasant odors like orange. Okay, so questions about diffusion. Okay, now I'm going to toss in a variable. Okay, we're going to move to osmosis. Okay. Okay, so let's say I've got the same situation here, okay, where I have a beaker of water, but I'm going to put a barrier in, okay, and you'll notice I'm showing it as a dotted line. Okay, this barrier, okay, is what we call a semi-permeable barrier. Okay, so semi means partial. Okay, permeable means you can get from one side to the other, you can permeate. Okay, so it means some stuff is going to get past this barrier and some things won't. Okay, so let's go ahead and drop my sugar cube in there. Okay, so I've dropped my sugar cube in and it starts to dissolve. Okay, but as it's ricocheting around, it can't get past this barrier. Okay, the holes are just too small. Okay, we'll make my holes smaller. for sugar to get past. Okay. So the sugar is going to stay on this side. It's not going to get to this other side. Okay. But with this semi-permeable barrier, okay, water can get past as it's zinging around, bumping into the sides of the beaker and into other water molecules. Some of them are going to zing over here. Okay, so they're going to be able to come over. Okay, now what we find when we see situations like this, when we run an experiment like this, what we see is the water level will go up on the side with the sugar, and it will go down on the side without the sugar. Okay, so the water moves toward the area of higher concentration of solute. Okay, we call that osmosis. Osmosis is the movement of water through a semi-permeable membrane. In response to different concentrations of solute. Okay, so it's going to move toward the sugar. Oops, I'm drawing on my equilibrium side. Okay. All right. So I'm going to give you two different ways of remembering why water moves toward the higher concentration of sugar. Okay. There's the human example. Okay. And then there's what's actually going on chemically. Okay. So sugar is having a party. Okay. And there's nothing going on over here in the apartment next door. Okay. So sugar's over here, they have music, they've got dancing, and water is over here going, I can binge watch this show later. I'm going to go over and I'm going to knock on the door and say, hey, can I join the party? So the water, I'll come over here to where the party is. Okay, does that help? Okay, let me show you what's actually going on. So I'm going to blow this area up so that we can see what's happening on a molecular level. Okay. All right, so I've got this barrier. Okay. And sugar. Okay, I'm going to draw a sugar cartoon here. And I'm going to draw some OHs here, because sugars have a bunch of OHs. And if I drop a sugar cube in, it is probably going to be sucrose, but I'm not going to draw a disaccharide. Okay. All right, so it's just too big to get through. Okay. It cannot squeeze through that opening. Water, on the other hand, is a much smaller molecule. Okay. So it can go in, the water here can go out. Okay, so in theory, it can move back and forth however it likes, okay, because it's smaller. But once it comes over here, it's going to hydrogen bond, okay, with the sugar, okay. It'll hydrogen bond with other water molecules, okay, but it's going to hydrogen bond, okay, with the sugar, okay. And they kind of hold hands, and once they start holding hands with the sugar, they can't get out into through these small holes until they stop hydrogen bonding, okay, until they bump into something and they get knocked loose from this static cling that is a hydrogen bond, okay. So that's why water tends to come in more than it comes out, is because it's hydrogen bonding with the solute. Yes, Amanda.
Okay, I'm just trying to understand this. It's a little confusing for me. Okay, so correct me if I'm wrong.
Sure.
osmosis is basically water going over to the sugar molecules because it's hydrophilic.
Right.
And then therefore it's can no longer go over to the other side because now it's too big.
Right.
Okay, so my other question is, what is that barrier?
Good question. So what is that barrier? So, which is an excellent lead in to our next topic. So when we do this experiment in the lab, this barrier would be dialysis tubing. Okay, so folks that have kidney failure, what we do is we take their blood and we run it through this dialysis tubing, and toxins can go to the other side and we can put their blood back in. Okay, so that's what we use in the lab when we do this experiment. Okay, but here's why we care. Okay, the cell membrane, okay, is a semi-permeable membrane. Okay, and so we see osmosis happening in cells. Okay, now here's the cell membrane. Okay, and it is semi-permeable. Okay, so that's why we care. Okay, so if we blow up my cartoon here, what we're going to see, okay, are phospholipids, okay, lined up in a bilayer. Okay. So the heads, okay, this round part, they are hydrophilic, okay, so this is philic, and this part here is phobic. Okay. And there is water on the outside of a cell, and there is water on the inside of a cell. So these hydrophilic heads, okay, have a negative charge, which means that they can interact with water. So I've got a negative charge all the way around here and all around the way here, but there is no charge in the center. Okay. So, okay, being a semi-permeable membrane, these phospholipids, just like the water and the sugar in my example, bounce against each other. Okay, so they're moving around. Sometimes they move far enough apart that water can kind of sneak on through. Okay, so water can go into a cell and it can go out because this is a semi-permeable barrier. Okay, how are we doing so far? But on the other hand, sugar, okay, it has all of these OH groups, and this slightly negative hydrogen, okay, just wants to hold hand, or slightly positive hydrogen, excuse me, wants to hold hands with these negative phospholipids. Okay, so they kind of get stuck. So even as things are moving around, one, they're too big to get through, and two, they're kind of stuck. Okay, so sugar molecules, come on, wake up, kind of stick to the outside, or if they're inside, they kind of stick to the inside. Okay, so water has a hard time getting through, but it can, okay, because channels open up occasionally. Sugar doesn't get through. Okay, so sugar cannot diffuse from one side of the cell to another. So if I have a bunch of sugar out here, the cell wants that sugar, it wants to eat that sugar, okay, but it can't get past the semi-permeable membrane. Okay, so we have more than just phospholipids, okay, in the membrane. We have transporter proteins, okay, that can let things pass this membrane, okay? But they check to make sure it's the right thing, and they make sure that, you know, once they get the sugar inside, they don't want it getting back out, okay? Once you buy those groceries, you don't want to just toss them in the yard. You spent good money on those. Okay, so you want to transport them into the house, into the refrigerator, and you don't want them to get back out. Okay, so we have these proteins that act as gates. Okay, so there are two different kinds of these transport proteins. Okay, some of them are passive. Okay, they just open up and things can come through. Okay, or they can go out. Okay, and so it's like, you know, just an open door swinging in the wind. Okay, people can come in and out, things can come in and out. Okay, and then there are active. Active, they grab a hold of what's on the outside and bring it in. Okay, and they take energy. Okay, so let's say that this one is a passive transporter. Okay, so we're going to color that pink, and it requires no energy. And we're going to color this one orange, okay, and it's going to be active. Setting up my legend here, okay? So let's say I've got a bunch of sugar out here, okay? I can just open up, okay, this channel, and the sugar is going to go down its concentration gradient into the cell, okay? That's diffusion, okay? But this channel has to facilitate, okay, the diffusion, okay? So facilitation is a fancy word for helping, okay? So this passive channel is going to allow facilitated diffusion of the sugar from its high concentration to a lower concentration inside the cell. Okay. Now, as that concentration drops, because I've facilitated diffusion, okay, we're going to have to switch to active transport. Okay, so there's not very many there. So this active transport protein is going to grab what few sugar molecules just happen to randomly bounce in and contact it, and they're going to grab it and they're going to hustle it on in. Okay, we call that active transport. Okay, how are we doing? I flipped your brains inside out yet? Okay, now this works with anything that is hydrophilic, okay? So it works with sugars, okay? Sugars cannot diffuse in and out of the cell unless it's facilitated by a passive transport protein, okay? They have to be actively transported, okay, by an active transport protein if there's low concentrations outside, okay, or if I've got a high buildup of toxins and I want to shunt them out, okay, that also works with salts, okay, which are composed of ions, okay. So you want a certain concentration of salt or ions inside of the cell, no matter what's outside of the cell, okay, for most cells. Okay, and this is what you need to know for this class. The concentrations of salts inside of the cell rounds to 0.1%. Okay, it's really 0.09%, but this rounds well. Okay, outside of the cell can be anything. Okay, all right, how are we doing? Yeah. Okay, let's talk about osmosis again. Okay, we talked about passive diffusion, okay, and the active transport. But let's say that this cell, okay, is living in lake water. And we're going to say there's 0% salt, okay? So I've got 0.1% salt inside, okay, the cell. And let's go ahead and make salt green. No, I want to make it blue. No, I don't. Yeah, let's make it green. Okay, which way is the water going to move? Since the osmosis comes into play here, which way is the water going to move?
Inside.
Very good. It's going to move inside because that's where the party is. Okay. Now, there's an experiment that I don't know if they do anymore. If you take human cells, specifically red blood cells, and they look at like, to me, they look like filled in lifesavers. Okay. And if you do the same thing, you drop human cells that have this 0.1% salt and you drop them into distilled water, okay? The water is going to flow in, okay? And this poor little cell is going to swell up like a balloon and then it's going to pop, okay? This is why we never give patients a distilled water IV solution because we don't want the red blood cells going pop, pop, pop. Okay, that would kill them. Okay, so bacteria, on the other hand, okay, can live very comfortably in a lake water. Okay, because outside of their cell membrane, they've got a cell wall. And it's rigid, okay? So as the water flows in, okay, as it starts pressing on the cell membrane, the cell wall holds it back. And it keeps the cell from popping, okay? And eventually there's too much water in here and the water stops flowing in, okay? And it's because of the cell wall. Okay, so you can't pop a bacterial cell, okay, if you drop it into distilled water. You're not going to see this happen, okay? But we humans, we don't have a cell wall. So if you drop red blood cells or any of our cells that are not protected by skin or your mucosa, they're going to pop, okay? Because the water flows in and it keeps flowing in. And there's nothing to hold back that pressure as it gets bigger and bigger and bigger. Okay, now what if we introduce this bacterium to an antibiotic that causes holes in the cell wall? What's going to happen? Any ideas?
So the water will come in then?
Yeah, the water will come in. Okay, so it's going to come in through these holes. Okay, and then what's going to happen? We've already got water coming in.
It'll explode.
It'll explode. Yep. it will lyse just like our red blood cells do. Okay. So just FYI, there's a whole class of antibiotics that attack bacterial cell walls, specifically peptidoglycan. Which is only found in bacteria. Okay. And so all of your psyllins do this. Okay. Penicillin, amoxicillin, ampicillin, methicillin, they all result in holes in this peptidoglycan cell wall, and they cause cells to lyse. Okay. All right. I'm going to clear the board just so that I have room. And I'm going to get my cells back up. Here's my bacterial cell. We're going to go ahead and give it an intact cell wall. And I'm going to get a red blood cell here. Okay, so just as a reminder, in both of them, I have, if I round, 0.1% salt on the inside of the cell. Okay, what if I did the same thing, did something a little different? Okay, what if I dropped it into seawater? Okay, depending upon which sea we're talking about, we'll say the ocean, we have approximately 2% salt. Okay, so we've got significantly more salt in this ocean water than we do on the outside of the cell, or on the inside of the cell, excuse me. Okay, so which way is the water going to go?
It will go out.
It'll go out, because remember, the party's out here. Okay, so on both of them, okay, more water is going to come out than is going in, because it wants to go to the area of higher concentration. And so my poor little red blood cells are going to shrivel up like little red blood cell raisins. Okay, and the same thing is going to happen, okay, to bacteria that are not evolved, not adapted to living in the ocean. Okay, so what's going to happen is that this cell wall can't shrivel up. Okay, but inside, my poor little bacterium is going to shrivel up inside of its cell wall. Okay, like a little bacterial raisin. Okay, because the water has been leaving. Okay, so it kind of dehydrates it. Okay? We call this in bacteria plasmolysis. Okay? You don't have to remember that term, but. All right, so what about bacteria that live in the ocean? We have a whole bunch of bacteria that live in the ocean very happily. Okay? So what happens is they have, whoops, let's change the color here. They have transporter proteins that actively shunt the water back in. So as fast as the water is going out, they move it back in. Okay? So that would be active transport. Okay? Our cells, okay, are not evolved to do that. Okay? They do move ions back and forth, and in A&P, you're going to learn more about that. Okay, yeah, we're not. That's why we do not give patients a 2% salt solution IV. Okay, so those of you who are in healthcare, what kind of solution do we give patients? We call it physiological saline. Anybody know what the concentration is?
Isn't it 0.9 or 0.09? I can't remember.
Yep, it's 0.09.
Okay.
Physiological saline, and it's usually sterile, so physiological sterile saline. But we can round it to 0.1. That rounds very easily to 0.1. But you're right, it's 0.09. So when we do that, water moves in and out at an equal rate. Okay. And so our poor little cells don't pop, nor do they shrivel up. And we call this an isotonic solution. Okay, something where it's higher outside is a hypertonic solution, and if it's lower, then it's a, okay, so my lake water example is hypotonic. Okay, so a good way to remember this, hyper is above. Okay, so it's like if somebody is hyperactive, they have above or more energy than the population as an average. Okay, if somebody is hypoglycemic, okay, their blood sugar has dropped. Okay, or if you give a hypodermic, okay, that is below the skin. Okay, so hypo is below, hyper is above, iso, and I forgot the O, is the same. All right, how are we doing? I know this is a tough subject. Okay, tell you what, I'm going to switch back to the PowerPoint, and I've got some questions for you to practice. Okay, what we've just learned. Okay, because that's a good way to study. So I'm going to pull a poll up. Okay, and ignore E. Okay, so let me move this out of the way. You drop a sugar cube into the center of a glass of water. Which area of the glass is down the concentration gradient? And yes, there is more than one right answer. Okay, we've got some good voting going on. Okay, we're almost to half, not quite. Okay, we are past half. Good job. Ooh, we just broke 75. See if we can get to 90% of it. Okay, we're at 80%. I need 4 more. Okay, I need 3 more. Okay, I think we've paused at 85%. That's all right. Okay, I'm going to go ahead and end the poll and share results. Okay. Now, with this type of question, what works best for me is to draw it out. Okay. So I'm going to go ahead and draw this out. Okay. So and on the test, you're going to be allowed scratch paper. Okay. So here's the water. Okay. I dropped the sugar cube in. Okay. So down into the glass where the sugar cubes rest, okay? Is glass hydrophilic or hydrophobic? When I put water into a glass cup, does the glass start dissolving?
Philip.
No, no, it does not start dissolving, so it is actually phobic. Okay, so can the sugar go into the glass? Can it go down into the glass? We'll go ahead and draw what this is. Here's the glass. And so can it go down into the glass?
No.
No, okay. So A is not the right answer, okay? So is down the concentration gradient into the center of the cube. And it's okay if you don't know the answer. That's why I'm quizzing you, is it's a good study technique. Okay, are more sugar molecules, as they're bumping into each other, are they going to get back into the elevator once the door is open? Yeah, no, they're not going to go back into the center. That's up the concentration gradient. That's swimming upstream. Okay. What about out toward the edges of the glass? Okay, is it going to go more this direction?
I would say yes.
Yes, you're correct. Yes, they're going to go more toward the outer surface. Okay, they're going to spread out. So that is down the concentration gradient. Okay, what about up toward the surface of the water? Is that down the concentration gradient?
I'd say yes also.
Yes, yes. So C and D are the correct answers. Okay. Now, if you picked one of the others, that's okay. We're still learning. Okay. And sometimes getting the wrong answer when you're practicing helps you remember better. Okay. All right. So now I'm going to switch this up. Okay. For the sake of repetition. Okay. I'm going to leave my cute little drawing here. So which direction is up the concentration gradient? Okay. Okay, we've got some good early voting. Okay, we just broke 50%. Okay, let's see if we can get to 75. Okay, we're at 85. Let's see if we can get up to 90. Okay, I think that's as far as we're gonna get. Okay. All right. So using my little drawing, okay, we're looking at the opposite of what I asked before. Okay. So which direction is going toward an area of higher concentration? Okay. So once again, okay, sugar can't go into the glass because it's not hydrophilic. So A would be incorrect. Okay, if you picked A, don't feel bad. We're practicing. B, toward the center of the sugar cube. That actually is correct. The higher concentration here is here. So we're swimming upstream. Okay, we're going against the trend. We're going up the concentration gradient. Okay, and these other two are going down. Okay, so this time the only correct answer was B. Okay. Any questions? Yes. Michelle.
Just a comment. I misread that question where it's saying down into the glass where the sugar cube rests. I didn't read that as the sugar going into the glass. I was reading that as down into the glass toward where the sugar rests.
Ah, gotcha. Yes. So yeah, that could be I should have been more clear on that answer option. So if it were on the exam, I would say, sure, if you're saying that that's going up the concentration gradient, sure, I'll give it to you. Because glass in this situation has two meanings. It means the cup, and it also means the glass that forms the cup. So it would be more clear on a test if I said that you dropped a sugar cube into the center of a glass cup of water. Yeah, so that's an example of, and yes, I did that on purpose, of, the discussions that we could have, during office hours after the exam closes. So sure, I'd give it to you. And then I would rewrite the question for later semesters so that it's more clear. Okay, Amanda.
Sorry, one day I'll figure out how to lower my hand immediately.
Oh, no worries. I wish they still had it on a timer because I do that all the time.
Well, I guess since I'm on here, I'm a little confused about the down and the up of the concentration gradient, because basically in your questions, they mean the opposite, because when your little example, they were going up when they were saying down. Oh, so it's meant to be that way.
It's meant to be that way.
That's not confusing at all. Okay, thank you.
No. And my apologies for the confusing terminology, but unfortunately, that's the terminology that we use. I think with the folks who developed this terminology of going up and down the concentration gradient, they were thinking of an analogy of a stream. Okay. If you have a high lake and you've got a stream running down from it, the high lake has more concentration of water, and the little stream is running down the concentration gradient. Whereas if you go upstream, and so I think that's what they were going for. But when you drop sugar or salt into a glass, a cup, down the concentration gradient is up. So for those of you who are fans of science fiction, going with Ender's Game, the enemy's gate is always down, and when you're in 0G. So when you are in a cell, okay, there is no up or down. Okay, it's either toward the higher concentration of solute, okay, which is up the concentration gradient, or down, okay, is away from the highest concentration. So my apologies for how confusing it is, but I can't help it. But that's why I use the sugar cube example, so we can discuss that and hopefully resolve that. I don't know if that helps. Okay, are you ready for another question? You're all going, no. Okay, let's go ahead and do a question with osmosis. Okay, so I'm gonna brine a piece of meat. Okay, it makes it more flavorful and it makes it more tender. Okay, So the concentration of salt inside of the meat, inside of those meat cells, okay, is 0.1% if I round it. And the salt concentration of the brine is 2%. Which direction is this water going to move? Okay, we've got some good voting going on. Okay, we're at 76%. Ooh, we just got to 80%. 85, 90. Yeah, I need 2 more. I think we're stalled at 90%. Okay, so most people picked out of the meat. That is correct. Okay, if you picked into the meat, that's all right. You're still learning about osmosis. This is a part of the process. So who would like to tell me why into the meat or out of the meat is correct? Yes, Amanda.
Because of your analogy earlier, because the party's on the outside. So it's going to go on the outside.
It is. It's going to go on the outside. Yes. Good job. Yeah, that's much easier than up and down the concentration gradient. Having a party is much better. Okay. Okay, now here's the same question, but I've switched it up. Okay, so instead of putting it in a salt brine, I have put it into a sugar brine. Okay, which direction is the water going to move? I did say which direction, right? Sometimes I say it backwards and I say, which direction is the sugar going to move? The sugar's not moving. Okay, we've got some good early voting. We were at 50% there for a while. Okay, we're at 80%. See if we can get to 90. Okay, we're past 90. Let's see if we can get, or past 80. Let's see if we can get to 90. Okay, so I'm gonna go ahead and share results here. Okay, so most people picked out of the meat. Okay, so why? The answer is B, by the way. Why is the water going to move out of the meat? Okay.
Because the sugar is 1% and the salt inside is only 0.1%.
Yes, very good. Good job, Elena. So it doesn't matter whether it's salt or sugar. Okay, it's still a solute, and the water is going to move toward the higher concentration of solute. Okay, good job. Okay, we went over the structure of the cell membrane. Okay, here is another question for you. Okay, since the function of the cell membrane is to act as a barrier, okay, keep the outside stuff out and the inside stuff in, why are they composed mainly of phospholipids instead of simple oils like olive oil? By the way, this is a harder question. Okay, we've got a few people voting. Because this is a harder question, it takes a little longer to read through it and think through the answer.
Does this have multiple right answers or is it only one?
There are multiple right answers. Yeah, but one of the right answers is a clear favorite. And by the way, if I put a question like this on the exam, it would be a multiple answer. Okay, we're at 85%. Okay, we're at 90%. Ooh, 95. I just need one more. Okay, I think I'm stalled at 95%. Okay, most people picked phospholipids have hydrophilic heads. Okay, so they can interact with the water inside and outside of the cell. Okay, true statement. Okay, that is why cell membranes, okay, are composed of mainly phospholipids, okay, instead of simple oils that don't have a hydrophilic head. Okay, all right, so C was the next favorite. It lost by quite a bit, but it's the next highest percentage. Okay. Phospholipids have hydrophobic tails that interact with each other, forming a waterproof barrier. Notice I put that in quotes, barrier. That is also a true answer. Okay. You've got to have that hydrophobic pore. Now, water can sneak past, but it doesn't do it really fast. Okay. So we do get osmosis, but it takes time. Okay, so both of these are correct answers. Good job. Okay, now you got to watch out for questions like these. I put these on purpose. Okay, so this one is a true statement. Okay, but it restates, okay, what I've already told you. It doesn't answer the question of why. Okay, so cell membranes are composed of phospholipid bilayers, but it doesn't answer why they're composed of phospholipid bilayers. Okay. So you got to watch out for answers that are true statements that do not answer the question. Okay. Now this one was intended to be funny. It's not terribly funny. I have better joke answers. So if you see anything that says, wait, it's supposed to be funny, reduce tension. But because people laugh at the joke answers, they want to pick it as the right answer because it made you feel good. So don't feel bad that you picked the joke answer. Also, the olive oil actually doesn't come from the cell membranes of olives, excuse me. Cell membranes of olives are phospholipids, okay? But olives pack a whole bunch of olive oil inside of the cells, okay, as food for the baby olive seed. Okay, and what we do is we squish out that simple oil. Yeah, just in case you're wondering. All right, let's go to the next one. Okay, we talked about transport in and out of the cells. Let's do another question, and I think this is going to be the last one. Okay, while you're voting, I'll check the time here. Yeah, this is about all we've got. Okay, now this one's tough. This is very tough, but it might help you with case study 1. And by the way, there's more than one right answer for this one too. Okay, we've got some good voting. Okay, we're almost to 50%. Okay, we're at 85%. Is that as good as I'm gonna get? There we go, 90%. Okay, I'm gonna end at 90. Okay, this is another one where we've got to draw, okay, what's going on, okay? And unfortunately, when I'm in the PowerPoint, I only have red. I don't know why. All right, so what I would do is I would go ahead and draw my membrane, okay, as a cartoon, and then to transport sugars across the membrane, okay? Oops, I didn't draw that very evenly, okay? All right, so I've got to be able to allow hydrophilic sugar to get from the outside that has water to the inside that has water. Okay, so I want to do this. Okay, now remember, we've got a negative charge, okay, through here on those phospholipid heads, and in the center we have no charge. It's hydrophobic. Okay, so hydrophilic side chains on the amino acids forming the center of the channel. Okay, do I have to be through here? Do I have to be hydrophilic? Okay, and the answer is yes. Okay, if they're hydrophobic, sugar is not going to go through there. Okay, because the hydrophobic side chains are going to keep the sugar out. Okay, so this is a true statement. This is correct. Okay. Oh, let's go with correct instead. Okay. So hydrophilic side chains on the amino acids on the inside of the cell. Okay, so this is inside the cell. Okay, this is outside. Okay, so would the side chain sticking out here have to be hydrophilic? Okay, yes, because otherwise the water would force it into the hydrophobic center of the cell membrane. Okay, so this answer is correct. Okay. Hydrophobic side chains on the amino acids in contact with the center of the lipid bilayer. Okay, so this part here. Okay, yeah, they have to be hydrophobic, otherwise they're going to shoot right out of the membrane. Okay, they're not going to stay there. So this one is correct. Okay. Hydrophobic side chains on the amino acids on the outside of the cell. Okay, we've got a ton of water out here. If this part was hydrophobic, it would shove it down into the membrane. Okay, so this one is incorrect. Okay, so things to think about as you are working, finishing up case study number 1, because I don't know, I might have a question about transmembrane proteins, proteins that go through the membrane. Okay, any questions? Okay, all right, I did want to have this really hard question so I could show you how to think your way through a question. Okay, draw a little model, okay, and use that model to figure out whether each of the answer options is correct or incorrect. Okay. All right, let's do past this. Okay, this is if we have time, practice for the test. Okay, and in the you can find similar questions in my practice quizzes that are in the study aids folder. Okay. All right, so things to do for next week. The embedded tutor assignment number 1, that's due by Friday. Okay, before Friday turns into Monday morning. Case study number 1 is due. Okay, so those are the things that are due by Friday. Okay, continue to work on the biology chemistry badge. Okay, that's due in a couple of weeks. Finish going over the chapter 3 lecture materials. Okay, and then because we've got a test coming up, it's going to open on Friday. It's going to stay open until a week from Saturday. So you'll have a week and a day in which to take it. Okay, so start up in your study. Okay, go back and review chapters 1 through 3. Okay, and then prepare for next week. We're going to start on chapter 4, which are eukaryotic cells. Okay, and write down any questions you have. Okay, any questions? Okay, if you think of any, okay, I do have office hours today at 1:30. Or email them to me. Okay. All right.